It's a question I think a lot of us wrestle with, isn't it? What really makes us us? Is it our genes, like the blueprint we get from our parents?
At the nature side of things.
Right, nature.
Yeah.
Or is it, everything else? Our experiences, the world we live in, nurturing.
Yeah, classic nature versus nurture debate. It's been around forever.
Identical twins, they offer such a unique window into this, don't they?
They really do, because identical twins come from just one fertilized egg that splits in two.
Meaning they have basically the same DNA.
Exactly. Virtually 100% identical genetic code. Think of DNA as this incredibly complex instruction manual.
Okay.
It's packed into the nucleus of our cells, this famous double helix structure. You remember A pairs with T, C pairs with G.
Right, the base pairs.
And it's amazing. You've got about 2 meters of this stuff crammed into a microscopic nucleus, all wrapped up tightly around proteins, forming chromosomes.
Ah.
And specific chunks of that DNA, those are the genes. They're like individual recipes for making proteins, maybe 20, 25,000 of them in humans.
So these genes tell our body what to do, essentially.
They direct everything. But here's the key thing. Genes aren't always on. They get switched on or off depending on what the body needs. That's gene expression.
Okay, so identical twins get the same instruction manual, the same DNA, but, and this is the crux of it, they don't stay identical, do they?
Not at all. Over time, you see differences emerge. Maybe one twin develops a certain health condition and the other doesn't.
Or they have different personalities, different habits, even look slightly different sometimes.
Precisely. So if the DNA is the same, what's going on? What causes these differences?
And that's really what we want to dive into today, isn't it? We're on a mission to explore why these genetic duplicates diverge.
And the answer, or a huge part of it, lies in this fascinating field called epigenetics.
Right, and how our environment, especially things like nutrition, these epinutrients, we'll talk about how they influence the way that shared DNA blueprint actually gets used.
Exactly. Genetics gives you the potential, the starting point, but it's definitely not the whole story.
So it's not just the genes. There's this whole layer of environmental influence.
A massive layer. Think about everything around us, the physical stuff, what we eat, toxins we might encounter, how active we are, diseases.
Okay.
Then the social side, parenting, culture, education, our relationships, and even our own internal world, our emotions, stress, trauma, mental stimulation, all of that interacts with our genes.
And that interaction point, that's epigenetics.
That's where epigenetics comes in, yes. It's
Yeah.
become a really hot topic.
Yeah, you hear the term a lot. But break it down for us. What is epigenetics, fundamentally?
Okay, so think of it this way. Epigenetics doesn't change the actual DNA letters, the ATCG sequence. Instead, it's about adding chemical tags onto the DNA or onto the proteins the DNA wraps around.
Like little sticky notes.
Isn't that a great analogy? Sticky notes or maybe dimmer switches. These tags tell the genes whether to be active, less active, or completely silent. They regulate gene expression.
Molecular switches, you called them earlier. So the environment, diet, stress, toxins, how does it actually flip these switches?
Well, those environmental factors can trigger the enzymes that add or remove these epigenetic tags. For instance, certain nutrients in our food provide the raw materials for tags like methyl groups. Stress hormones can send signals that lead to changes. Toxins might cause specific tags to get stuck on.
Right, so for twins, even though the DNA code is the same,
Their different life experiences mean they can accumulate different patterns of these epigenetic tags over time.
so one twin might have a gene switched on while the other has it switched off because of something they experience differently.
Exactly. And that could explain why, say, one twin gets type 2 diabetes and the other doesn't. Even if they both have some genetic susceptibility, their environments led to different epigenetic profiles.
So it's like they have the same sheet music, the DNA, but epigenetics is like different conductors interpreting it based on life events.
That's a really good way to put it. It's a second layer of information, an interpretation layer shaped by life. And these differences, they can start incredibly early.
How early are we talking?
Even in the womb.
Really? But they share the same womb, don't they?
They do, but it's not perfectly uniform in there. Think about placental position. One twin might get slightly better blood flow or nutrient delivery.
I never thought about that.
Yeah, or slight differences in oxygen levels or exposure to the mother's hormones. Tiny variations, but during that critical development phase.
They can have bigger consequences down the line.
Potentially, yes, affecting things like metabolism or maybe subtle aspects of brain development, setting slightly different paths right from the start.
Wow. So the divergence begins even before day one outside the womb. And then, obviously, life happens.
Life happens. They develop unique interests. One loves sports, the other prefers books.
They have different friends, different teachers, maybe.
Different social experiences shape their emotional development, their communication styles. Maybe one twin has a really great math teacher early on, gets encouraged, and develops a real knack for it.
While the other twin had a bad experience and avoids it.
Exactly. Small choices, different reinforcements, different paths taken, they all add up, making them more distinct individuals over time.
It really underscores that constant interplay, doesn't it? That dance between nature and nurture.
It's absolutely central. Our environment dictates how our genetic potential gets expressed. You might have genes linked to, say, depression, but they might only get activated epigenetically if you experience significant stress or trauma, which maybe only one twin does.
Okay. And the flip side.
The flip side is potential that needs nurturing. Someone might have a genetic predisposition for musical talent, but if they never get access to instruments or lessons.
That potential might never fully blossom. The environment didn't provide the right signals.
Precisely. It's a partnership between genes and environment.
So when we think about something as complex as personality, how does this play out? How much is baked in and how much is learned or experienced?
Personality. That's a big one. Twin studies have been hugely informative here.
Like the Minnesota twin study.
Exactly, that's a landmark one. They found strong genetic links for traits like general cognitive ability IQ scores tend to be quite similar.
Okay.
Also activity levels, even some basic aspects of temperament, food preferences, mannerisms, lots of similarities, even in twins raised apart.
Which points strongly towards genetics, right?
It does, but then you look at other things. Their specific knowledge, their social skills, their taste in music, political views, religious beliefs.
Much more variation there.
Much more. Those seem much more shaped by their unique experiences, their upbringing, their culture. The Minnesota study showed this too. Even separated twins differed significantly in things like moral reasoning or cultural values.
So it's definitely not all one or the other. It's this complex mix.
A very intricate mix. And epigenetics seems to be the mechanism or a key mechanism mediating that interaction.
Okay, let's get into those mechanisms then. You mentioned these chemical tags, these molecular switches. Can we unpack that a bit more? What are the main ways genes get switched on or off epigenetically?
Absolutely. Let's break down the main players. Probably the most studied is DNA methylation.
Methylation, okay.
This involves attaching a small molecule, a methyl group. It's just CaSO, 1 carbon, 3 hydrogens, directly onto the DNA molecule itself, usually onto a cytosine base.
And what does that do?
Generally, it acts like a stop sign. It often silences the gene, making it harder for the cell's machinery to read it and make a protein from it, like putting tape over that part of the instruction manual.
Got it. So methylation often means off. Are there specific enzymes doing this?
Yes, they're called DNA methyltransferases, or DNMTs. They're the ones adding the methyl tags. And interestingly, problems with methylation patterns are linked to diseases like cancer.
How so?
Well, sometimes cancer cells use methylation to wrongly silence genes that should be active, like tumor suppressor genes, the ones that normally put the brakes on cell growth.
Okay, wow. So methylation is like a silencer. What about turning genes on or making them more active?
For that, we often look at histone modification. Remember the histone proteins DNA wraps around?
Yeah, the spools for the DNA thread.
Exactly. Well, those histone proteins themselves can be chemically tagged. Common tags include acetyl groups, methyl groups again, but on histones this time, and phosphate groups.
And these tags on the histones, what do they change?
They change how tightly the DNA is wound around the histones. Adding an acetyl group, for example, acetylation, usually loosens the winding.
Making the DNA easier to access.
Precisely. It opens up the chromatin structure, making the genes in that region more available to be read. So acetylation often means on. Enzymes called HATs, histone acetyltransferases, do this.
Okay, so HATs turn genes on by acetylating histones. What turns them off?
Removing those acetyl groups does.
Yeah.
That's deacetylation, done by enzymes called HDACs, histone deacetylases. This tends to tighten the chromatin back up. Adding methyl groups to certain spots on histones can also tighten things up and silence genes.
So methylation can happen on DNA or histones, but acetylation on histones is mostly an on signal.
Generally speaking, yes. There are enzymes that add and remove histone methyl groups too, HMTs and HDMs. It's a complex system of marking the histones to control DNA accessibility.
It really sounds like a sophisticated control panel. Methylation on DNA, modifications on histones. Anything else?
Oh yes. There are also non-coding RNAs and cRNAs.
Non-coding, meaning they don't make proteins.
Exactly. Unlike messenger RNA, mRNA, which carries protein recipes, ncRNAs have other jobs. They act as regulators.
How?
Well, you have small ones called microRNAs, mere RNAs. They can bind to mRNA molecules and basically block them from being translated into proteins, or even cause the mRNA to be destroyed.
Like little sensors.
Sort of.
Yeah.
Then there are long non-coding RNAs, LNC RNAs. These are bigger and can do various things, like helping to reshape the chromatin or guiding those histone-modifying enzymes to the right genes.
So RNAs that aren't making proteins are busy managing the ones that do.
In many cases, yes, they add another layer of control.
Okay. And you mentioned chromatin structure itself. How is that managed?
That falls under chromatin remodeling. This involves specialized protein complexes that use energy, ATP, to physically shift the nucleosomes, those DNA histone units, around.
So they can slide them along the DNA.
Slide them, push them out of the way, or change their spacing. This directly controls whether the DNA in a particular region is accessible or not.
So you have tightly packed regions
Heterochromatin. Genes there are generally off.
and loosely packed regions.
Euchromatin. Genes there are generally accessible, ready to be turned on. These remodeling complexes are constantly adjusting this landscape, like librarians rearranging shelves.
Okay, that makes sense. Was there one more mechanism?
Yes, a related one. Histone variant replacement. Cells can swap out the standard histone proteins for specialized versions called histone variants.
Like different types of schools.
Kind of. These variants, like H3.3 or H2AZ, have slightly different properties. They can change how stable the nucleosome is or how other regulatory proteins bind, adding yet another way to fine-tune gene expression.
Wow. It's incredibly layered and complex. DNA methylation, histone modification, non-coding RNAs, chromatin remodeling, histone variants, all these epigenetic layers controlling the basic DNA code.
It's a dynamic system that allows cells to respond to signals and adapt.
And you mentioned earlier this idea of an epigenetic clock. How does that tie into all this?
Right, the epigenetic clock. It's fascinating. Researchers found that certain patterns of DNA methylation change quite predictably as we age.
Across the whole genome.
Yes, at specific sites. By measuring the methylation levels at these sites, they can estimate a person's biological age.
Biological age. How is that different from just how old you are?
Your chronological age is just the number of years you've lived. Your biological age reflects the functional state of your cells and tissues, how old you seem on a molecular level, and they don't always match up.
So your biological age could be older or younger than your birthday candle suggests.
Exactly. And twin studies show that while identical twins start with very similar epigenetic patterns, their epigenetic ages can drift apart over time.
Because of different lifestyles.
It strongly seems so. Things like smoking, poor diet, chronic stress, lack of exercise, they appear to accelerate epigenetic aging. Your biological clock ticks faster.
But healthy habits might slow it down.
That's the hope, and emerging evidence suggests it. A healthy diet, exercise, stress management, these might help keep your biological age more in line with, or even younger than, your chronological age. It means our lifestyle is literally influencing how we age at the DNA level.
That's incredibly powerful. Which brings us right back to diet. You use the term nutripigenomics. Tell us more about how food talks to our genes.
Nutripigenomics is precisely that, the study of how nutrients and bioactive compounds in food directly influence our epigenetic patterns.
So food components can add or remove those sticky notes we talked about.
They can. Many nutrients provide the essential building blocks for epigenetic marks. We mentioned methyl groups earlier, folate, which you get from leafy greens, liver, vitamin B12 from meat and dairy, choline from eggs and soy, betaine from beets and spinach. These are all crucial methyl donors needed for DNA methylation.
Okay, so they provide the ink for the methylation marks.
In a way, yes. Then you have compounds like sulforaphane found in broccoli and cabbage, which can inhibit those HDAC enzymes we talked about, the ones that remove the on switches from histones. So sulforaphane might help keep beneficial genes active.
Interesting. What else?
EGCG from green tea seems to modulate DNA methylation. Omega-3 fatty acids from fish influence histone modifications and gene expression related to inflammation. Polyphenols in berries, dark chocolate, tea can activate protective genes.
So it's not just about calories or basic vitamins in the old sense. It's about specific molecules in food sending signals to our genome.
Exactly. Diet is a major environmental input that constantly shapes our epigenome. And differences in diet are a big reason why twins might have different health trajectories.
Which leads to this idea of epinutrients. What defines an epinutrient?
Epinutrients are simply those dietary components that play key roles in supporting healthy epigenetic processes.
Like the methyl donors you mentioned.
Yes, like folate, B12, choline, betaine, they donate methyl groups, but also compounds that support enzymes involved in adding or removing tags, or that influence those non-coding RNAs, or support detoxification pathways that clear out potentially disruptive chemicals.
Can you recap some key epinutrients in good food sources, just to hammer it home?
Sure, so folate, leafy greens, liver, legumes; choline, eggs, soy, meat, liver; vitamin B12, meat, fish, dairy, fortified foods; betaine, beets, spinach, whole grains; sulforaphane, broccoli sprouts, cabbage, kale; EGCG, green tea. These are some heavy hitters.
It really makes you think differently about your plate, doesn't it? That broccoli isn't just fiber and vitamins, it's potentially tweaking your gene expression.
It absolutely is. Food is information. Your dietary choices are constantly writing and rewriting notes on your genome.
And this has implications beyond just our own health, right? You mentioned the phrase, you are what your grandparents ate.
Yes, there's growing evidence, particularly from animal studies, but also human observations, suggesting that the epigenetic patterns influenced by diet, stress, or toxin exposure in one generation might actually be passed down to the next.
Passed down. How?
It seems some epigenetic marks, especially methylation patterns, might escape the reprogramming that normally happens during egg and sperm formation. So the experiences of your parents or even grandparents could potentially leave subtle epigenetic traces in you.
That's mind-boggling. Is there strong human evidence for that?
The Dutch famine study is probably the most cited example in humans.
Right. You mentioned that earlier. Tell us a bit more.
So during 1944-45 in the occupied Netherlands, there was severe famine. Pregnant women were getting maybe 400, 800 calories a day.
An incredibly stressful, nutritionally deprived environment.
Extremely. And researchers looked at the children who were conceived or in early gestation during that famine decades later. They found these individuals had higher rates of obesity, diabetes, heart disease, even mental health issues compared to siblings born before or after the famine.
Wow.
And crucially, they found differences in DNA methylation patterns in these individuals, specifically in genes related to growth and metabolism that were still detectable 60 years later.
Suggesting the prenatal famine environment left a lasting epigenetic mark that influenced their health decades later.
That's the strong implication, yes. It's a stark example of how potent environmental influences can be, especially during critical developmental windows, and how long-lasting the effects might be.
And potentially passed on. How far could this epigenetic inheritance go?
That's still an active area of research. Some studies suggest effects might span 2 or 3 generations. Estimates vary, but maybe around 60% of methylation patterns could have some heritability, influencing perhaps over 1,000 genes. Animal models show clear evidence of transgenerational effects from diet or stress.
So the way we live today, our diet, our stress levels, might not just affect us, but could echo in our children or grandchildren's biology?
It's a distinct possibility. It adds a whole new layer of responsibility to our lifestyle choices, doesn't it? It's not just about us.
It really does. It feels a bit daunting, but also empowering.
Mhm.
Because if our choices influence our epigenetics,
Then we have some degree of control. We're not just passive victims of our genes or even our ancestors' experiences. We can actively shape our epigenetic health.
okay, so let's talk about that. What are the practical takeaways? What can people listening do to support a healthy epigenome?
It comes back to the foundations of good health, really, but viewed through an epigenetic lens. First, nourish your genes. Focus on a whole foods diet, rich in those epinutrients. Lots of colorful plants, leafy greens, berries, healthy fats, quality protein sources like eggs, fish, legumes. Minimize processed foods, sugar, things that might be epigenetically disruptive.
So eat real food, basically.
Pretty much. Second, move your body. Regular physical activity isn't just good for muscles and heart. It sends positive signals to your genes, activating pathways linked to resilience and longevity.
Okay. Diet and exercise.
Right.
What else?
Manage stress. Chronic stress is a major negative input for our epigenome. So finding healthy ways to cope is crucial. That could be meditation, yoga, spending time in nature, ensuring good quality sleep.
Sleep is important for methylation, you said.
Yes, sleep is critical for many restorative processes, including maintaining healthy epigenetic patterns. So prioritizing sleep hygiene is key. It's about creating an overall lifestyle that sends health-promoting signals to your genes.
Right. So putting it all together,
Yeah.
our DNA is the script we inherit.
The basic
But
text.
epigenetics, heavily influenced by our environment and especially our diet, those epinutrients acts like the director, the editor, maybe even the lighting technician.
I like that, yes. Deciding which parts of the script get emphasized, which get muted, how the whole story is presented.
And the message for everyone listening is that those choices we make every single day, what's on our fork, whether we go for that walk, how we handle stress, they matter profoundly.
They really do. They're shaping your health right now at a molecular level and potentially influencing the biological legacy you pass on.
It's a powerful thought to end on. So maybe the question for you, the listener, is this. How are your daily choices writing your genetic story today? And what kind of legacy are you building, epigenetically speaking, for the future? Definitely something to chew on.
Absolutely. It opens up a whole world of possibility for taking charge of our health.